So before we get started, I wanted to give you an official welcome to the virtual March of Dimes High School Science Week. My name's Monica Wirt-Parkinson, and I'm the Director of Public Programs here at the Salk Institute. High School Science Day is one of our very favorite traditions here at the Salk Institute, and in fact, this is the 31st annual event. We were very sad we couldn't welcome you onto campus this year, but we're really proud to continue the event in this virtual space. All of our Salk researchers are so excited to chat with you today, and they're really looking forward to answering your questions. So, the Salk Institute is actually named after Dr. Jonas Salk. You may have heard his name very recently in the news. He was actually a key player in another public health crisis, the polio epidemic, that affected many children in the 1940s and 50s. Dr. Salk was the person that developed the first safe and effective polio vaccine. He then went on to pursue another dream, which was to create a collaborative environment where researchers could work together to explore the very basic principles of life. 60 years later, the Salk Institute is a world-renowned basic research facility. The Salk Institute is actually known as a modern architectural treasure, and it was recently named as the most beautiful building in California by Business Insider. And this photo particularly is a very famous photo of the Salk Institute courtyard. And it features our main fountain feature that runs down the middle of the travertine courtyard and appears to flow directly into the Pacific Ocean. This fountain is named the River of Life, and it's actually meant to symbolize the knowledge of the Salk Institute trickling out into the world. Twice a year on the equinox, the sun sets down the middle of the fountain, which is this photo that you see right here. And it's a really famous photo that photographers set up and wait hours for that they can make sure that they get a picture of. Just a quick overview of today's format. We're doing a quick introduction right now, and then we'll move into our virtual lab tour. Following the lab tour, we'll have a great panel discussion. So we really want to make sure that you submit your questions. I'm going to go over in just a minute how you do that if you haven't already put one in. Each video is a standalone experience with unique content, and we definitely encourage that you attend more than one lab tour. You may discover a topic that you didn't think you were interested in, but it may spark an interest with you. So if you get a chance, we definitely encourage you to attend a couple of different lab tours. All of the links were sent out in the registration, and they'll be made available after the event is over in pre-recorded format and will be sent out to all registered attendees. We always hope that everything runs really smoothly, but if there's any technical issues, you can email us at education@salk.edu and just be a little bit patient with us while we fix our technical issues if we run into any. So the March of Dimes High School Science Week is our favorite part is interacting with you, and we really want to know what you're curious about. So we'll be using your questions to guide the panel discussion. It's really simple to submit a question for our panelists, and you can submit to an individual panelist or you can submit to the panel and the lab overall. All you have to do is visit the submission form that's located in the description right on the page that you're looking at. And if you're curious about our panelists, you can click on the Show More button, which will lead you to their bios as well as additional information about the lab's research. Really quickly, before we get into today's lab tour, we wanted to go over a couple of other high school science programs that we have. One of the programs that has been in existence for many years is the High-Top-Brody High School Summer Scholars Program. It's an 8-week program with a biotech bootcamp and 7 weeks in the lab mentored by a Salk scientist, and it is a paid internship. It's a really unique opportunity to conduct real-life research and develop science communication skills. You also have an opportunity for industry visits, seminars, and special events with your cohort. We're really hopeful that the program will take place this summer in 2021, but it's very dependent on public health guidances and protocols. So keep checking our website. We hope to have a different program status updated for you in March. The program that we are going to be hosting no matter what is titled the Introduction to Research Science and Communication Virtual Program. It's a really fantastic 5-week professional development opportunity open for students all throughout the country. You do not need to be a San Diego resident. You just need to be enrolled in high school in the 2021 high school year. It's 5 to 10 hours per week, mostly asynchronous, and we've taken a lot of the program elements from the High-Top-Brody High School Summer Scholars Program. So you'll learn how to read journal articles, analyze data, prepare a scientific presentation, and you'll attend virtual labs. It all culminates with a final capstone project on a topic of interest. And it'll definitely be happening in the summer of 2021. In fact, our program lottery is currently open. It closes on March 31st. So if you're interested in participating, we definitely encourage you to visit the website. If you have any questions or you want to apply for the programs, you can email us at education@salk.edu or visit our website. All of the program information is loaded up on the website. And before we start today's lab tour, we just want to say a brief thank you to Ann and Neil Blue High School Science Fund. It's really thanks to the Blue family, as well as all of our supporters, partners, and volunteers, including the March of Dimes, that make Salk Education Outreach possible. We're really excited today to have you visit our plant molecular and cellular biology laboratory. It's led by Dr. Joanne Chory. This video will give you a glimpse into what a day in the life of a scientist is like in the lab. Each video was designed and filmed by the lab, so you'll have a chance to see a unique point of view of the lab. It's a really great opportunity to learn a little bit more about our plant molecular and cellular biology laboratory, as well as the work they're doing around the Harnessing Plants Initiative, which is a larger initiative on the Salk Institute campus that is really driving to use optimal plants and their natural ability to capture and store carbon with the goal of helping to solve climate change. And with that, I welcome you to sit back and relax and enjoy the virtual lab tour. We'll be joining you again in about 10 minutes for the panel discussion. Thanks so much.
Our environment is constantly changing. The sun rises and falls in a day, and the seasons change over a year. In addition to this natural cycle, increases in carbon dioxide and other greenhouse gases caused by human activity in the burning of fossil fuels is increasing global temperatures, bringing more extreme and unpredictable weather patterns. These changes to our environment affect all life on this planet. In the Plant Molecular and Cellular Biology Laboratory, led by Dr. Joanne Chory at the Salk Institute, we seek to understand how plants sense changes in their environment, how they respond to these changes, and ultimately, if plants can be part of the solution to our climate crisis. To study plant responses to the environment, we take advantage of different plant species. For example, the mustard weed, Arabidopsis thaliana, is small, easy to grow, has a short life cycle, and a small, compact genome, which is easy to mutate and genetically modify. This species is the workhorse of the plant biology community. Basic principles which we learn in this plant can then be translated to other crop species on which we work, such as tomato, oilseed and vegetable varieties of brassica, and rice. Other more unusual species, such as the carnivorous plant Venus flytrap, are used to learn about how plants evolve mechanisms to sense specific environmental stimuli, such as in this case, the touch of insect prey, and how these touch signals are converted into an electrical signal that propagates across the leaf to induce movement and prey capture. This electrical signaling is a feature shared with other plants in response to herbivore-induced wounding. For much of our work, we need to carefully modify a plant's environment and track its growth and response. In our climate control facility, we can modify the wavelengths and intensity of light, the humidity, and the temperature at which a plant grows to simulate real-world conditions. Further control of the environment in which we grow our plants can be achieved by growing seedlings in tissue culture conditions, which also facilitates easy measurements of plant root and shoot architecture.
One of the goals in our lab is to understand the molecular basis by which a plant senses and responds to these environmental conditions. What are the genes involved? To address this, we screen through mutant libraries for plants that have altered growth patterns or conduct experiments in which we track changes in gene expression as a result of an environmental stimulus. When we have identified the DNA sequence of the gene of interest, we can further manipulate the plant genome to test our hypotheses. Like other plant molecular biology labs, we take advantage of specialized bacteria that naturally live in the soil, with which we can introduce foreign DNA into the plant genome. Using this system and gene modification tools, we can introduce specific lesions into the DNA or express modified proteins that can be visualized on a microscope. Using fluorescence microscopy, we can image in which tissues a particular gene is expressed, and where within the cell the protein might localize and function. This can give us clues as to the biological processes within which a gene and its protein product are involved. Lastly, plants with modified genomes are returned to different environments, and we carefully track how they respond. Key manipulations may be replicated in crop varieties to further test if we can make predictable changes to crop plant growth in specific environments. Ultimately, our methods determine the basic genetic underpinnings of how a plant grows in diverse environmental conditions. The next big challenge being addressed by the Salk Institute is to see if we can now use plants to alter our environment on a global scale and reduce the levels of the greenhouse gas carbon dioxide in the atmosphere.
Now that you've seen what we do at Salk, I'm going to show you a little bit about our off-campus greenhouse. Follow me! Welcome to our greenhouse. To accomplish this goal of combating climate change through plants, we need to increase their carbon sequestration ability, which is their ability to suck up carbon from the atmosphere. Here you can see we grow a variety of plants. To do this, we have a state-of-the-art 12,000 square foot greenhouse where we grow our crop plants and study them for indication that they'll be able to sequester more carbon. In order to make sure our watering is controlled, we have an automated irrigation system. The water flows through the pipes here and through the emitters out into the spitters and into the plant pot. We are constantly monitoring the amount of nutrients or water that a plant leaches out. Now let me show you our lab. Here we have a freeze dryer, a plant drying oven, a growth chamber, a biosafety hood, water bath station, an autoclave, and our X-ray machine. Continuing on, we have our scanning stations, a workstation, our refrigerator, and our imaging cart. One of the most important things we do in the greenhouse is process the plant samples that we have grown. Phenotyping the plants is one of the most important aspects of the project, as it allows us to identify what we want to move forward with. When the plants are ready to be processed, we will excavate the entire plant from its pot and wash the root system. The first step in this is to dump the pot in a vat of water and rinse the roots of the substrate they've been grown in. Once the roots have been thoroughly cleaned, we will then dry them out and sieve the water that we use to wash the roots in order to capture any remaining roots that may have fallen off in the process. Once the plants have completely dried out, we will then weigh the samples and collect data on the roots and the shoots. Depending on the crop, we'll dry the plants in a multitude of different ways. For most crops, we air dry, hang dry, and then put them in our drying oven for a certain duration depending on the crop type. Depending on our needs, the plants have one of two fates. They will either get processed for data, or we will seed bulk them so that we can retain seed for future studies. When seed bulking Arabidopsis thaliana, we will bind and stake the plants once they have bolted and are creating flowers. When they've reached the appropriate growth stage, we will allow them to dry out completely, and once they are dried, we will harvest them. Once the Arabidopsis has sufficiently dried, we will cut the base of the plant and remove the stake and the bag and the plant altogether. Then we'll take the plant out of the bag and we'll put it in a sieve. The plant is then crushed in the sieve and we separate the excess dried plant material and the seeds at the same time. One problem we run into with studying the roots is that you generally have to excavate and kill the plant to get data on the root system. In order to study the root system without killing the plant, we have an incredible piece of equipment that x-rays the root system and generates an image of inside the pot without having to excavate the plant. The machine slowly turns the plant and takes approximately 3,000 images while the plant is turning. It's technology like this that propels us forward in our research and allows projects like the Harnessing Plants Initiative to be possible. In order for a project like this to be successful, we have to work with a diverse set of individuals in order to solve our global issue. After all, global issues require global, scalable solutions. Ultimately, at the Salk, we are working hard to address how plants sense and respond to their environments, and if in turn we can take advantage of the power of plants to tackle climate change for a better planet.
We really hope that you enjoyed a chance to take a peek into our labs. We are so excited. We have some great questions lined up, but first I wanted to take a moment and have each of the panelists introduce themselves. So why don't we start with you, Anne-Marie?
Hi, everyone. My name is Anne-Marie, but you can call me Annie. I am a research assistant as part of the Harnessing Plants Initiative, and currently I am studying a microRNA to try to increase root biomass in our model plant, Arabidopsis thaliana.
Thank you. And McKenna, if you want to go next.
Yeah. Hi, guys. My name is McKenna Hopwood. I'm originally from San Diego, and I am the greenhouse operations lead. I manage the 12,000 square foot greenhouse that we have that we grow all of our study plants in.
Great. Thanks, McKenna. And Carl.
Good afternoon, everyone. My name's Carl. I'm a staff scientist in Dr. Chory's lab. My background, I'm kind of a general geneticist by training. So I did my undergraduate back in Australia, where I'm from. I then moved to New York City, where I did my PhD studying neurobiology. And now in Dr. Chory's lab, I study plant responses to the environment. And I'm particularly interested in more unusual exotic plants. So for example, the carnivorous plants you saw in the video, I study their particular adaptations to catch, to sense, catch, and digest insects.
Great, thanks so much. Just a reminder, please feel free to submit questions all throughout the panel. We're going to try to get to as many as we can. The first question that we have in is, what do you find most inspiring about your work in the Chory lab? And Carl, I think we'll start with you.
Sure. I mean, it's a pretty amazing place, I would say. You're surrounded by very talented, very intelligent people, and this helps drive the science forwards. You know, there's people like to think big and tackle big problems, and the Harnessing Plants Initiative is an example of that. So you have few limits, and you've got a lot of freedom to explore your own interests as well as Joanne's interests as well.
I think that's great. That's a really great way to tie it back to Jonas Salk's original vision too. And Annie, why don't we hear from you next about what you find most inspiring about your work in this lab?
Yeah, I think the most inspiring thing is being able to use these natural systems in place to help us get through the world and figure out how to best leave our impact on this world.
Thank you. And McKenna.
Yeah, I think the most inspiring thing, I think, is just the fact that we might be able to make a global impact with our project. And, you know, if we're able to get the science right behind it, then we have the potential to really make an incredible impact throughout the entire world. So definitely inspired by that.
It's a very inspiring motivation that you all have working for this lab. A very kind of similar question is, what skill have you learned in the lab that you're really the most proud of? And McKenna, if you don't mind answering that question first.
Yeah, I'd say communicating science is one of the skills that I'm always trying to work on, and doing events like this is always making it easier for me to do that. So I like being able to communicate science and increase science communication around projects. So I've really enjoyed being able to do that and share kind of what the Harnessing Plants Initiative does and what we as scientists do every single day in the lab.
Yes, and we thank you for being a part of this too. Having you be a communicator of science is just so fantastic. Annie, what's a skill that you're most proud of that you've learned in the lab?
So I've been in this lab since my undergrad. So this lab really helped build me up as a scientist. And I think I'm most proud of finally being able to figure out how to work with RNA because it's so unstable. And now I'm working with measuring small RNAs, and just getting into the whole RNA world has been a lot of fun.
Carl, what about you? What's a skill you've learned that you're most proud of?
I've been in science, in biology now for almost 20 years. And in that time, I think the biggest change, which I've seen in molecular biology, has been the use of big data sets, and particularly, so these are enormous data sets, and they often use a lot of DNA sequences. And so something I've had to teach myself in this lab is computer coding to try and analyze this data and make sense of it. And I think I speak for a lot of scientists of my generation who would have thought, as biologists, that we would end up being programmers as well. So I'm proud of that. it's something I've had to tackle and address in my time in this lab.
Yes, and that's a huge skill and so applicable to all areas of science right now, the computational side. So we have a really great question next, more on the science side. Is it possible to apply the same process of optimizing a plant's natural ability to capture and store carbon to marine plants such as kelp and algae? And I think, McKenna, are you comfortable answering that question?
Yeah, so one distinction between algae and actual true plants is that algae isn't actually a plant by the true definition of plants. So we are tackling or trying to tackle a part of the Harnessing Plants Initiative is the coastal restoration aspect of things. So we do have a team of scientists who are working on the true plants, the true marine grown plants. So just a good indication for this, if you are swimming in the ocean, a lot of the marine plants will flower. And that is a good indication that that is indeed a plant and not an algae. So we are working on part of this as the HPI is increasing coastal restoration areas. All of the coastal restoration areas, wetlands, those are really important aspects of our climate and our ecology in order to increase carbon sequestration. So wetlands, kind of all of that stuff that are on our coastal areas are really, really valuable for carbon sequestration and fighting climate change. So this is an aspect of the project that we are trying to tackle, and we recognize the importance of it, absolutely.
Great, thank you so much. So this is a question probably for all of you because you're in varied positions here in the Salk Labs, but what level of education do you need to be in your particular position? And maybe we'll start with you, Carl, please.
Sure. So I'm now a staff scientist. I just got my promotion this week, actually. For my level, you do need a PhD to stay this far in academic science. I'm not a professor, but I'm at a position where I can apply for my own grants and lead my own research within Dr. Chory's lab. So you would need a PhD. That said, and you'll hear from others, not every position in science do you need a PhD, but on the academic track, you would, by and large.
Great, thank you. And Annie, how about you?
I think for a research assistant position, most research assistant positions would require a bachelor's degree, but there are instances where you can get into a lab during or before your undergrad, before you get your bachelor's degree, and work your way up into a research assistant position.
Great. And McKenna?
Yeah, for mine, just a bachelor's degree is required for managing the greenhouse. I do have a lot of say in the science and I'm able to do experiments on my own, but definitely not to the scale that Carl can. Additionally, kind of same with what Annie said, that you can work your way up to positions. So if it's not necessarily a degree, but also experience is highly valued. So if you have a degree on top of a few years of experience, that is a great way to start.
Great, thank you. And then our next question is, why is increasing root biomass important? Is there anyone that wants to take that question in particular?
Yeah, I'll take that question.
Great. Oh, go ahead, Amy.
I figured I'd answer it since that's what I'm working on. But yeah, increasing root biomass is important because we want to essentially create this larger vessel in which that we could put more multi-carbon molecules in. And why the roots specifically is because the roots are in the soil, which is a carbon sink for us. So eventually, once the roots break down, they will seep further into the soil and enrich the soil that way.
Great. Carl, did you have anything you wanted to add to that question?
No, not really. I mean, but Annie summed it up. I mean, larger roots is more carbon. So the more carbon you can get down into the soil and hopefully trap there by generating particular versions of carbon which are recalcitrant to breaking down, the better we'll all be.
Great, thank you. So the next question I have for you is, what challenges have you faced while on your journey in science? Because I think you all took very different paths and probably had different experiences. So maybe, Carl, if you could chime in first about what challenges you might have faced on your journey through science.
There's always challenges at every step of the way. Some are personal challenges. My life has seen a lot of, life is unpredictable. This year, for example, coronavirus, who would have thought that? I've had health issues in my families to deal with. So there's the personal challenges. There's science at a high level can be very competitive and aggressive, but it can also be very exciting. So, you know, all these things are balanced. So the challenges for me in a large part have been personal, and then also just dealing with people. Everyone's different. Everyone has their own personality. It's not good and bad necessarily, but you have to be able to deal and communicate with these people and come to agreements. Science is very collaborative, so you need to be able to work with others.
That's very helpful. I think that applies to all career aspects that people may be considering right now, as communication is definitely key. McKenna, what challenges have you faced, if any, on your journey into your current path in science?
I'd say just trying to kind of follow where the path leads me, sort of thing. There's been a lot of different opportunities that I've had that, you know, are those kind of forks in the road that you don't really necessarily know are the forks in the road at the time. And so it's just figuring out what's best for the situation based off of kind of what Carl was touching on, personal aspects of your life as well as your professional development, and just kind of navigating that world so that you are making the best decisions for your future. And also working with people. I think a big misconception about science is that it's kind of all on your own, but there's so much working with people and collaboration that just learning how to work with these different personality types has been a welcome challenge, I'd like to say.
Thank you. And Annie, do you have anything you want to share about any challenges you may have faced on your journey through science?
Sure. A lot of my journey happened during my undergraduate. So some of the challenges I faced was maintaining my grades, schooling, extracurriculars. I had a lab assistant job at another facility, and then I did some volunteer work at the Salk Institute. And so it was a lot of things just up in the air that I had to juggle with. And eventually I realized where my priorities were, and that I eventually just wanted to be in an academic setting. And then learning what can I do to optimize my time in an academic lab.
Thank you for sharing some of those challenges and how you face them. I think that will be great advice. The next question that we have for you is a really great question. What do you think will be the best plant for absorbing carbon? What do you think it could be, and why? I'll open up to anyone who wants to chime in.
Can we throw in a hypothetical plant here? I mean,
yes.
We've already discussed deep roots. So that's one. Another, which we're looking at, is this idea of the kind of molecules which this carbon is being stored as. So some molecules will degrade faster in the soil simply because bacteria have greater access to them chemically, and others will degrade at a slower rate. And then also, by necessity, I think it's fair to say that a lot of the plants which we're looking at currently are annuals. But ideally, long term, and there's groups around which are addressing this issue as well, is to look at perennial plants. Can you turn our crops tend to be annuals, so we harvest them each year. But if you can have the plant stay around and grow for a much longer period of time, you're not always cutting it down, releasing that carbon, but you get it to stay fixed in the plant. So that's something else which I think can be addressed as well.
I think it's also important to consider the amount of space one of these plants will take up. So a lot of the reason we're focusing on the crop plants is because they take up so much space worldwide that we can really make the biggest impact with that.
Thanks. That's very helpful and interesting as how you're approaching that particular problem. So this is kind of a question I think about work overall is, does lab work ever get monotonous or long?
A few chuckles from us here. Who you vote aye, raise your hand. I mean, does someone else want to answer?
Yeah, I mean, I would say it's more monotonous. You can correct me if you guys disagree, but at the lower levels of lab work that you're doing, generally, if you're a lab tech, you tend to be the one that does the really monotonous work. But as you kind of grow and advance in your career, you're able to make more research-dependent decisions and able to kind of design the experiments you want to launch and so on and so forth. But there's definitely aspects that get monotonous. That's why it's so important to be passionate about what you're studying.
Yeah, and a lot of, to continue on from that, a lot of this monotonous work, right, is to get towards this goal, which is really exciting, which is the result. We work towards the result. So you have these moments of monotony, but then you have this periods, however brief they might be, but these periods of excitement. Where you kind of jump up and down in your seat when you're staring at the computer and you see that result you were hoping to get. So it's punctuated periods of monotony and excitement.
And you can definitely get balance in your work. I sometimes will try to do some reading in the morning, and then I'll go down to our facilities and hang out with my plants for a little while, tie up their inflorescence, as you've seen McKenna do in the video that we showed you earlier. But yeah, you can definitely balance out your work in between being in front of a computer, being next to your plants, doing some reading, doing some writing. There's a lot of aspects to being a scientist that make it not so monotonous.
Absolutely. I think you all kind of hit on finding something you're passionate about is probably the biggest driving factor. So finding that thing that you can stick through the monotony to have those great moments of excitement. So this next question is really interesting, and it may have changed depending on where we've been the last year during the pandemic. But the question is, what does a day in your life look like? And McKenna, do you want to share first, since you're in the greenhouses? You're outside the lab a little bit in a separate space.
Yeah, that's a great question. So I usually start off my day by lapping the greenhouses, doing pest control watches, checking that what insects have come into the greenhouse, what we have to deal with that day, if anything needs manual watering, if we have kind of anything that's been missed watering, just doing general checks of the greenhouse to make sure everything's going well. I do have a lot of computer work, so unfortunately I do spend a good chunk of my time in my office just answering emails, organizing experiments in meetings, kind of all of that stuff. And then just general maintenance and depending on what we're doing that day. So if we're harvesting plants, if we're phenotyping plants, if we're potting plants, really just assisting where I can help out. So we have a large team of individuals at the greenhouse that really make the greenhouse what it is and keep the greenhouse running. And I'm just kind of trying to make sure that it continues on with the help of these individuals. So a lot of difference. Not one of my days is the same, which is part of why I love science, is kind of it's always changing. But yeah.
Thank you. And Carl, what about you? What does a typical day in your life look like when you're in the lab?
I think similar to McKenna. Firstly, I should say COVID-19 has changed my typical day. I'm currently primary daycare for my daughter at home. So you adapt. So I tend to look after her at the day during the week. And then I go in and work evenings and weekends now. So when I get to lab, the first thing I do is the essentials, similar to what McKenna said. So for me, that's watering plants. It's doing a lot of tissue culture. So a lot of my more exotic plants, my exotic monster plants, I propagate through tissue culture. So a lot of tissue culture, and then it's whatever experiment needs to be done, whether it's protein work, RNA work, DNA work. That's what would usually come next, and that's usually the experiments I'm doing and hoping for those exciting results which I was talking about.
Great, thanks so much. And Annie, did you have anything you want to add to that?
Yeah, I do a lot of bench work. There's COVID has provided a lot more opportunities for reading and writing, for sure, and some days are just data collection days. So you put on a good podcast and do some data collection. But yeah, there's a lot of moving parts, but not every day looks the same.
Great. Thanks so much for sharing that with us. So this is a great overall question. What prompted you or inspired you to get into the field that you're in? And Annie, do you want to start with that, please?
Yeah, so I, after high school, went to community college, and I went to this really great community college where most of my science classes were attached to a lab class, and we were doing a lot of hands-on work. We got to stain different bacteria. We got to actually work with real human cadavers. It was a really wonderful experience, and was an opportunity to cement for me that science is where I wanted to be. And then my first job as a lab assistant, that's where I got to realize scientists are really cool, really weird people, and they're the kind of people I want to hang out with and work with potentially in the future. And they've been a real big motivator for getting into this field.
Great, I'm glad to hear that. What about you, McKenna? What sort of inspired you to go into the field that you're in?
Yeah, I've always really been interested in science. It's been kind of a continuous thing throughout my life. I started in college with a business economics degree. I was a year as a business major, and I knew it was not the path for me. Again, still super interested in science. It was actually a trip to Yosemite where I was on a hike, and I kind of realized like I need to do something involving this. So my degree is in environmental science. And so I, that summer switched my degree and started studying science and really haven't looked back. So once I graduated, I worked in a plant biology lab, and that really sparked my interest in plants. And then from there, I went to a research greenhouse setting where I really got to know the plants in and out and was really interested in how we could experiment with them and discover new things through them. And then my path led to here. So been more involved in kind of the science and ag aspect of things, which I think is really interesting, but it was kind of a walk around to get to where I'm at right now.
I think that's a really great lesson, that you're not always tied into the first thing that you decide you want to major in college, and to be very flexible and open to following your interests. What about you, Carl? What prompted you to follow this career path?
I feel I'm giving the same answer as McKenna on this one. I actually started with a double degree with law. I thought I would be a lawyer. And I'm not going to say any lawyer jokes here because that might be inappropriate. But I always grew up loving science, and that was where my passion lay. And so I ended up dropping the law and continuing with science. And for me, I'm not married to plants. I've studied both animals and plants. I like to have some application to what I do. It's nice. And all my projects have some application, whether it be the HPI or crops or even some sonogenetic applications with my carnivorous plant work, looking at molecules which can respond to touch and putting them in mammalian systems. So that's nice, but it's also not essential for me. I just think that the natural world around us is beautiful. It's awesome. There is so much beauty out there in nature, and if you can see that, I think I think that the world is a great place because of this. So that's what drives me. It's to understand all that beauty which I see around us, the bizarre and wonderful things which go on in our natural world.
Yeah, thank you. I love hearing about some of your roundabout ways to get into the career that you're in. So another great question is, besides harnessing plants, what other projects is the Chory Lab working on right now? And Carl, if you want to tell us a little bit, I want to hear a little bit more about your work with carnivorous plants. I think it's so interesting.
Sure. It's one of my projects. The Chory Lab really made a name for itself. Joanne started off doing a lot of pioneering work on plant hormones and plant responses to light. So that was the specific environmental variable which she started working with and has broadened out from there. The carnivorous plants was something I started for education outreach and kind of evolved into a much larger project. So here we're looking at how plants respond to touch. So all plants respond to touch to some degree. You can imagine a plant blowing around in the wind, and actually plants in a very windy environment, they tend to have thicker trunks, tend to be a bit shorter, so they sense this mechanical stress. Something like a carnivorous plant like a Venus flytrap has a very rapid response to touch. So this becomes a unique system in which to study this kind of sensory modality in plants. and to see a very fast change. So that's one of the unique, and they're also cool, right? I mean, they eat bugs. They turn the tables from what we expect. So they're cool, but we also learn some fundamental aspects of plant biology. And as I alluded to before, perhaps some of what we learn we can also start to use in different, more applied systems.
Great. Annie or McKenna, do you have anything else to add about other projects outside of harnessing plants you might work on?
Not particularly. No.
Thanks.
It's muted, but we're both pretty much harnessing plants people.
Yeah. Okay, so this is a great question that kind of also plays off of what you do every day in your work as a scientist. Do you do more of designing experiments or data collection or data analysis when you're approaching your work?
It really just depends what stage you're at in your project. Some days require more data analysis. Other days you're just planning the experiment and seeing how other people have done it and figuring out what the best way to do it for whatever it is that you're trying to test for. So it just depends.
I think go ahead, Carl.
I was just going to say, by and large, I would say for most of us, hopefully I'm speaking for most of us, if not, correct me, but I feel most of our time is done doing the actual experiment. I mean, you can design an experiment. Let's say you want to generate this particular type of plant, either through transgenics, so putting a foreign piece of DNA into the plant, through mutation, or through a cross, a traditional crossing of different strains together. So you can design that experiment in a day or a couple of days, depending on how complicated it is and how much thought goes into it. But then it can be months to actually generate that plant. So by and large, most of what we do goes into the actual doing of the experiment. Then there's the data analysis, and that can take quite a bit of time too, and the planning. So I think that would be a fair summary of what we do.
Thanks. Yeah, that's really helpful.
I agree with Carl on that, and it also matters about what you're experimenting on. So some experiments we have, 250 plants, some we have 2,400 plants. So for those ones, we're obviously spending a lot more time collecting data because you have to go to each individual plant and collect the data for it. And generally speaking, at least in the greenhouse, because there's so much replication, once we have one or two designs, we can utilize those designs kind of over and over again to launch more experiments. So yeah, definitely agree with Carl.
Thanks, that's really helpful. So this is a really interesting question. Have you looked into non-vascular plants such as mosses as an efficient carbon converter?
Well, mosses don't have roots, I don't believe. And so the whole point of us looking into the vascular plants that we are looking into is the fact that they build roots, and we can use the roots as a way to force carbon into the carbon sink, which is the soil. So, it's an interesting question, but I can imagine that if we were to try to use mosses, that we would end up losing more CO2 in some way back into the atmosphere because they don't have roots.
That makes sense. Thank you. So one more quick question. If you could tell us, how do you collaborate with your colleagues around the Mesa? I know you're so close to the Scripps Institute of Oceanography and you have UCSD right across the street. So what does it look like to be a scientist, and how much does collaboration play into your role here while you're conducting your research?
Maybe I'll jump in first. I do a lot of collaboration, and it's something which the collaboration just popped up in my almost 10 years here at Salk. I collaborate with a group, very talented group, of mechanobiologists at Scripps Research Institute, not the Oceanographic Institute. So this is a lab which can do things which we just can't. They do electrophysiology. They look at little electrical impulses in mammalian cells. So they've been able to look at some of our plant proteins And so we've had a very good collaboration with that group. I also collaborate and do some teaching at the University of San Diego. So they grow a lot of the plants there, and this is a way to engage undergraduates in some of the activities which we do as well. And again, they've got different expertise than me. They can do some chemical techniques which I'm not trained and equipped to do. So these collaborations have been ways to bring together people with different talents and different skill sets and build projects and stories which wouldn't otherwise have been possible. So that's my story on this particular question.
Thanks for sharing. Annie or McKenna, do you want to share anything else about collaborating?
Yeah, I think a big thing is that most science is built upon science before it. So a lot of what, when we're planning our experiments, we'll do a lot of research and read a lot of papers based off of people who have done similar things, and then often reach out and get advice and just talk to them about, the science behind it and why they did this certain methodology. So a lot of even just preliminary designing research and your experiments involves a lot of talking and communicating with other scientists who have done something similar to you.
That's great. Thank you.
Yeah, I would say for collaboration, just try to be brave about it. It is a little bit scary to reach out to these people who are more senior and more experienced than you, but it's the best way to learn, honestly, is to figure out what it is that you don't know from these people who can help you answer your questions.
I think that's some great advice. So we have a great question, more general, just any words of wisdom or advice for high school students that are out there considering a career in science that you would wish to share with them. And Carl, do you want to go first?
Sure. Don't be scared of math. I think, as I said on one of the first questions you asked me, who would have thought I would be a computer programmer starting out in biology 20 years ago? But this is a big part of what we do now, certainly in molecular biology and in a lot of biological sciences. The data sets are just becoming enormous. We have all this computing power behind us. We have new techniques coming up all the time. So don't be afraid of this. Keep up with, study something like statistics, you don't have to do computer programming per se, but to be at least comfortable with the jargon and the terminology, and don't be scared of it. So try and keep up some maths. That would be my number one recommendation to people interested in biology research nowadays.
Thanks. That's great advice right now. McKenna, what about you? Any advice you want to share with high school students?
Yeah, I think finding your passion is really important to figuring out what you want to do. And it's just as important to figure out what you don't want to do as what you do want to do. So, there's never wasted time in you trying new things out. Additionally, it's way easier to say no than to say yes, and you will be told no a lot of times. But if you're passionate about something and you find the right person, you just need one person to say yes, and then you're on your path to where you want to go. So just if you're passionate about something, you will be able to find something and find support for that thing, especially if that kind of comes out in what you do. And just stay true to kind of your passions and what you want to do.
That's great advice. Passion and perseverance. And Annie, what about you?
Yeah, just try to maintain a good work-life balance. That's been a big topic of conversation among scientists recently, because it's so easy to just get lost into the science that you kind of forget to take care of yourself. And so, ways to do that. Make sure that you're eating well, you're cleaning up after yourself, you're doing your laundry. Because when you've taken care of yourself, you can go into the lab feeling peaceful and ready and well rested. And that just makes for better research and better science.
I think that's great. That's applicable to everyday life, even just getting through high school right now, too. So we have a final question for you. And what is something that you've learned in the lab that's changed the way you live your life? So does anyone want to go first with that? I know you might need a minute to think about it.
It's kind of a curly question, isn't it? I do think it is the people interactions. So that's going to be my answer to it, because you do meet, you meet different people in science, and part of it is working with these people to do interesting science, to get results and build stories. But everyone's a little bit different. So and as I said, it's not good and bad, but it's fun. It can be a challenge sometimes, but working with other people will help you at home, interacting with people more generally. So this is something which is always a learning process, and something which I've learned from my time in the lab.
Thank you. Annie McKenna, is there anything that you've learned in the lab that sort of changed the way that you live your life now?
Yeah, I've learned that freaking out doesn't help in any situation. just like I mentioned before, working with RNA and how fragile it is, and sometimes we work in wells that are like super, super small, and so you have to keep track of everything that you're doing, and the best way to do that is to calm yourself, to breathe, to find your center, to like lay out all the materials that you need and the instructions that you have, and remaining calm. A lot of lab work has taught me how to like calm my emotions and center myself. And I feel like I brought that into my personal life, and I wouldn't have been able to do that if it wasn't for having to force myself to be calm for various experiments.
That's a great skill set you've developed through your experimentation. McKenna, is there anything you want to add?
Yeah, I think mine touches back on what Annie said last time. I have learned a lot about the science behind mindfulness and mental health, and how that kind of impacts the rest of your life. And it's not necessarily directly related to the plant lab, because as you can probably all tell, we're very interested in a lot of different things. But just knowing the science behind how taking time and taking care of yourself really impacts your life, and really just makes you a happier person in general, has been one of the biggest learning things that I have just infiltrated into the rest of my life over the last 4-ish years. But it's been really cool to see how that impacts things and impacts works and relationships, as well as the science that we do. So just taking care of yourself and slowing down also allows you to not freak out if things happen. So kind of just taking the time to have that work-life balance.
Great. Well, thank you so much for all of you that submitted such fantastic questions. And thank you to Carl, McKenna, and Annie for sharing such great advice, telling us a little bit about your journey, and talking a little bit more about the science behind the plant molecular and cellular biology lab. We really appreciate your time, and for producing such a great video. We had such a great time watching the video. And with that, thank you so much for joining us for this panel discussion for the March of Dimes High School Science Week. We look forward to seeing you again tomorrow. Thanks so much. Bye.
Thank you.